Hi, science friends. This is a bit of a shorter lesson today, so I want to start for just a second with a little bit of a review to kind of hit the highlights from yesterday. You should be able to draw a diagram that represents how alternative RNA splicing can code for two different proteins. So if this is our gene here, you have exons, so we'll call it exon 1, exon 2, exon 3, exon 4, exon 5. And those introns have already been sliced out in post-transcriptional modification, but you see that we have exon 3, exon 4, and exon 5 that could make one protein. You could have exon 1, exon 4, and exon 5 that make another protein, and lots of different combinations thereof. And so how we splice that RNA and put those pieces together to translate them can make lots of different proteins. And we see that in the human genome and how we can get so many different polypeptides synthesized from a relatively small amount of DNA. Okay? So that is alternative RNA splicing. The next part, how is a protein targeted for degradation and what cellular component is responsible for degrading it? So it is targeted by ubiquitin. Ubiquitin is the molecule that is put on its surface that's going to mark it. And then a proteasome is what is actually degrading it. Basically some enzymes that kind of cut and chop it up into little fragments. And so that happens at the end of the protein's life or at the end of like translation or something like that. Okay? So what I want to talk to you about today is embryonic development and genetic programming. So we know that we all start out as one zygote, a sperm and an egg come together, and that zygote starts to divide. And in that process of division, we know that it's giving rise to identical cells with the exact same genome. And so through the process of cellular differentiation, that is how those cells become specialized in structure, but also in function. If you look at different cells or different tissue types, they look different. A nervous cell looks very different from a skin cell. And so the process of differentiation is what makes the organs different, makes the cells different. We also have a phenomenon known as morphogenesis, where you start to create a form in that. And so how do all of these things happen? Well, they depend on the genes that an organism expresses and the proteins that it produces. And so the expression and the production of those proteins basically goes back to gene regulation, which is what we've been talking about extensively the last couple of days, having to turn on certain processes and turn off certain processes. And so in the egg that you get from your mom, you have cytoplasmic determinants in it. So those are substances that the mom provided in the egg. It can be RNA, it can be proteins, but they set up gene regulation in these first few cells. And so when this first cell divides, you get different cytoplasmic determinants in each of the cells. And the combinations of those cytoplasmic determinants determine what cell type will be formed. Okay? And so there's signals that the cells receive from other cells that neighbor it. So remember in our cell signaling unit, we talked about paracrine signals. And so these are paracrine signals. They're called inductive signals that come from other cells that cause a change, though, in gene expression. So there's a specific type of paracrine signaling called inductive signaling because they cause that change in gene expression, and it stimulates a specific signal transduction pathway that causes a cell to develop in one way or another. Okay? So these signals can come from contact with cell surface markers like you see in this diagram. binding of growth factors that are being secreted by neighboring cells. Lots of different options. They can come from proteins that are expressed by the embryos in their own genes or passed down from the mother. And so these signals help determine differential gene expression. Okay? And so this process that leads to differences in the cell is referred to as determination. Turning genes off, turning genes on, turning these genes off and on that are called master regulatory genes. So when it's, this master regulatory gene, MYOD, which helps to develop a myoblast, which is in muscle cells, gets turned off, then it's not going to develop those muscle-specific genes. But when it gets turned on, then you will develop those muscle-specific genes and to be able to form a muscle cell. Once a cell has undergone determination, it's irreversible, and so it can't change from a muscle cell to something else. Even if you put it in a different location, it can't. It's going to express specific genes for specific proteins that are only found in that cell type, and have a specific structure and function for that cell type. All right. So some of the genes that are responsible for this are called homeotic genes. So homeotic genes determine pattern formations. So basically what pattern formation means is determines how things develop. So, like in a lot of insects you have like an egg stage and then an embryo stage and then a larval stage, like a tadpole that goes into a frog. You have the egg and then the larval stage, which is like the tadpole, and then the frog stage. And so those types of pattern formation is controlled by a special type of gene called a homeotic gene. If you have a mutation in a homeotic gene, that is generally going to be what we call an embryonic lethal. And so a mutation in any type of gene that's super important like that is going to cause death at that stage. In humans, we would see that as like a miscarriage. Generally, miscarriages are going to happen in genes that are like really, really important. Okay, and so that's kind of sometimes what we would see. That's an embryonic lethal because you've had a mutation in a very important and relevant gene that's not going to be able to allow that organism to continue to develop. Maternal effect genes, those are genes that are passed on from the mother. Now, maternal effect genes are also often embryonic lethal. So if they have a mutation, then it will cause death at the embryonic stage. That organism will not go on to develop. But a maternal effect gene is basically kind of an epistasis, so to speak. And so whatever the mother has, that overrides the offspring. So if a mother has a dominant, or if the offspring has the dominant trait but the mother has the recessive trait, a maternal effect gene will kind of dominate in that condition. And so that it masks the offspring's genotype, and it's going to be the genotype of the mother regardless of what that is. So that's just kind of a category of genes. And then the last thing I want to talk about here is morphogens. So morphogens to help determine the axis. So it determines left from right. It determines which end is going to be the head end, which end is going to be the tail end, which side is the dorsal is the back side, the ventral is the belly side, and so it helps to determine body axes. Also, mutation in those genes are also going to be an embryonic lethal because, again, these are really important genes. Genes for pattern formation are really important. And so that's all I've got for you today, okay? Not a whole lot of really testable material on here, but I just want you to see that gene expression is regulated at every level and kind of how that plays out, just a basic brief overview. So we have one more lesson to wrap up this unit, and we will do that tomorrow. But for today, I want you to go ahead and complete the gene expression personal progress check. There are about 8 sections that we have kind of covered in this unit of gene expression. But one of them that is grouped in gene expression is biotechnology, and we have not hit that yet. We will do a separate mini unit on that following our gene expression unit. That's what's coming up next for us, is our separate biotechnology brief unit, and then we'll move on into evolution. But if you get stuff on biotechnology in your personal progress check, realize that I'm coming back to that. That's coming up next, but I couldn't not assign that part for the personal progress check. So just kind of guess on those questions that are real biotechy, if they're about gel electrophoresis or anything like that, and we will get to that information. But most of the stuff, I think there's 24 questions, you should be good to go on. All right. If you have any questions, let me know, and if not, then you have a great day.